So now back to the protein-protein interaction map, and we had these drugs and we had these compounds that we think could have antiviral effects. We didn't have the virus propagating in San Francisco at the time, but we leveraged our large network that Jacqueline alluded to and collaborated with scientists at the Pasteur Institute in Paris, in particular Marco Fagnuzzi, and then Adolfo Garcia Sastra at Mount Sinai Hospital in New York. And they were able to test to see which of these 69 drugs and compounds that Kayvon and Brian and Jack had predicted, which ones had antiviral effects. Many of them actually did, most of them did. And interestingly, 26 of these drugs are presently being tested in clinical trials for COVID-19. We're monitoring many of these but from afar, but we're also intimately involved in a couple of these. I just want to briefly go through with you. Here's different groups that came out of this work, but two translational regulators. First, zotadifin. This is a translational initiation inhibitor. This is a connection that came from the MAP. It's from the company Effector. Kayvon Shokat's one of the co-founders. This has potent antiviral effects. The FDA just approved the clinical trial for this drug for COVID, and DARPA is actually funding it. So we're very interested and excited about this one. I want to spend a little bit more time on this other drug called Aplidin. There's also a connection that came from the MAP. It targets a translational elongation protein called EIF1A. This is a drug that's actually being used to treat multiple myeloma from the company PharmaMar. And here is a structure of the compound. It's called Aplidin when it's used to treat multiple myeloma, plitidepsin when it's used to treat COVID. And here's a structure of it binding to its target EIF1A. This wasn't from us, this is from previous work. Interestingly, this compound is coming from this sea squirt, which lives exclusively off the coast of Spain, off of Ibiza. I hear that's a big party place. I want to visit that at some point. But that's an interesting kind of tidbit from where this compound comes from. And we've actually been involved in a lot of the screening and gotten access to a lot of the screening data from collaborators around the world, looking at different drugs and compounds to see what effects they have on SARS-CoV-2 infection. This one is by far the most potent. Here we're looking at HEC293 cells expressing ACE2. It's got a sub-nanomolar IC90 value, and in this cell system, it's 30 times more potent than remdesivir. When you go to primary lung cells, you can see Aplidin is very potent as well, again, at a much lower concentration than remdesivir. And this work, all of it was done in collaboration with Chris White and Adolfo Garcia Sastra. Adolfo's got mouse models where we can test as well, where you can, with an adenovirus, express human ACE2 in mice. And then you can infect those mice with SARS-CoV-2. You're adding drugs over time and then extracting the lungs and looking at the viral titers. And you can see with Aplidin or plitidepsin, you get about a 500-fold decrease of infection. And again, much more potent than remdesivir at a much lower concentration. And at the time, we had said this could potentially have value. Back in March. We started to collaborate with the company PharmaMar, based out of Spain, and they were carrying out a phase 1, phase 2 clinical trial for COVID. This was completed a few months ago. The data looks fantastic, and we're actually writing a paper with them right now, which we're hoping to submit next week. And because of our work, very excitingly, a couple weeks ago, Aplidin has entered phase 3 clinical trials for COVID-19 at 27 different sites across 12 different countries. So we're hoping that we're all going to hear some good news about this drug, and this will be another weapon in the fight against COVID-19. And I just really like this because it's connected back to the proteomic work that we had carried out initially. And the fact that we could move so fast is also a testament to the collaborations that went into making this happen. And just a few words about the variants, which we're all obviously worried about. And we're studying these in great detail, these different variants. The point here is if you target a host protein, you don't have to worry so much about resistance, drug resistance, because we don't mutate as fast as the virus, right, obviously. And I would argue the virus would never mutate enough to overcome its reliance on that particular host protein. So if you're able to target SARS-CoV-2, it doesn't matter how much SARS-CoV-2 will mutate, the drug should still have an effect. And consistent with that, plitidepsin is just as potent against the UK variant as the original lineage, and you see the same thing against these other variants as well. So I think this is a great advantage of targeting a host when you're looking at trying to target SARS-CoV-2, but not just for that virus, but for the next pandemic and all viruses as well. I'm going to come back to some of our work looking at the variants in just a few minutes.
Right, it's sort of, I think we all kind of know that, and I should probably read the paper you all wrote as well. But I'd be curious to know the details of the PharmaMar story and Aplidin. Is that, did I say that correctly?
Aplidin or plitidepsin, it's called when it's used to treat.
So that's in phase 3 clinical trials. A lot of people, it sounds like, have worked on getting that drug into place. selflessly for the most part. Someone's going to benefit, probably PharmaMar, probably not the scientists, except maybe for being on a paper. And it seems as though what you're doing as well is harnessing resources to support the community and support independent researchers, which is a great effort. But it seems like there's a systemic problem with scientific enterprise, like you mentioned. But is there an effort for things like the PharmaMar development to help change the system?
Yes, I mean that's a very good point. I mean, thinking about the pandemic we're in, obviously, PharmaMar made the drug. They own the drug, and if there's any value there, they're going to benefit from it, and probably as they should. But I think that's just a kind of a representation of one disease. I think we should be doing this for all diseases, right? I mean, to me, this has shown the power of these proteomic approaches to uncover novel targets that have great therapeutic value. And I kind of embarrassed we haven't been doing this before. Is the pandemic out of fear just forces you to kind of say, oh, what can we do, and who can we do it with, and how fast can we do it? And for me, it was realization is, well, we're closer than we think, that not just in the academic world, but in the industry world and the pharmaceutical companies and the academic world. So for me, this has been an amazing set of experiences where I've learned from, and now I want to apply that to many other diseases. So I think that there's great value in learning from what we've done here and applying them to different diseases. And I'll say there's a lot of effort now going on to come up with new infrastructure that would be kind of a hybrid between academia and pharmaceutical companies. Okay, so I think on both sides there's a realization we need new options. And I think we're going to be, many groups now are looking at this, and because of this pandemic, we're going to have let's just say a new way of doing things. I don't know, Jacqueline, did you have anything to add to that?
There is another question by CS1 from Spain. Would you like to come up online and then ask your question? Okay. The question goes like this. In Spain, a phase 2 study will begin with Aplidin, 122 COVID-19 patients without hospital admissions with a single dose of 7.5 mg. Is it better to have a single dose of Aplidin in one day, or is it better to distribute the dose over several days?
Yeah, I mean, that's a great question. This was approved 2 days ago as phase 2. This ESC1, I think this is a name I'm familiar with. I think he's on Twitter. He's been following us in Aplidin and Plitidepsin and PharmaMar. So, I mean, thanks for pointing this out. In the other clinical trials that I alluded to, it's twice, right? It's 2 treatments, 1.5 milligrams and 2.5 milligrams. And they've started this as a single dose at 7.5 milligrams. I don't know. It's not clear to me, but I'm super happy they're doing this as well. So we're monitoring this, and I think ESC1 will be probably emailing me or tweeting me the results before I know them. So I appreciate your interest in all of this, and we're excited to see what's going to come out of all these clinical trials around the world.
Would you like to come up and talk to Dr. Krogan, ECS1? If you're still there.
Would you like to reveal yourself?
Oh.
That's okay. We can keep by social media. That's great.
Good.